Morning, everyone. I'm Al Toops, Senior Agronomy Field Sales at Biome Makers. Thanks for joining us today on this webinar. What we're going to talk about is opportunities and values that we're going to be able to show by using the Biome Makers technology. Biome Makers is a AgTech company. It was founded in Silicon Valley in 2015. We're a global leader in setting the standard in soil health with our BeCrop technology. So that's some of the things we're going to go through and show you how you can use this technology to better enhance your soil management practices. Today on the panelists joining me are my colleague at Biome Makers, our senior agronomist, Jacob Parnell, and then Devin Clarke from Yara is with us today to talk about the project we did there at the America Incubator Farm in Modesto, California. Thank you guys for joining us today.
Thank you, Al.
If this is the first time you've seen one of our webinars or heard about Biome Makers, you may be wondering what Biome Makers is. So to begin with, we'll talk about some of the important questions that I have that maybe you may be thinking about. BeCrop Technology is a proprietary DNA sequencing intelligent computing workflow powered by the largest global taxonomic database of over 10 million microorganisms. The BeCrop technology uses the soil microbiome as a bioindicator of soil functionality, which breaks down into three main categories. We're looking at soil quality with a soil sample, the health of the soil, and the crop growing in that soil, and the nutritional values where we really kind of hone in on the nutritional nutrient pathways and how they're affected by the soil biology. BeCrop is also used to verify input products and performances, and we make these recommendations and predict the effects of products on yields. Today's webinar, we'll hear from firsthand experience from Devin on a project we did there, showing how we use this technology in a management practice setting in the field to investigate how intensive fertility management impacts the soil biology in almonds in a system that we looked at in California. So after we go through this experience and overview of what we did there at that trial, we'll have a minute or two at the end of the meeting, the end of the webinar, to have questions and answers and be able to ask any questions you have. Just let our panelists know, and we'll respond back to those. So this session is recorded, and we'll provide you a link that we'll email before the end of the week, and you'll be able to go back and look at the recording again. And with that, I'll jump right in, and we'll talk to Devin and Jacob and let them introduce themselves.
Perfect. Jacob, do you want to go first, and then and I'll jump into my section?
Sure. My name is Jacob Parnell. I am, I guess, my background is in microbial ecology, so I've done a lot of studying bacteria, particularly in soils. I've been involved with microbiomes for about the past 14 years or so. And so what I really like to do is I really like to see, based on the microbial patterns, the shifts that we see in the soil, you know, how that's basically giving the soil different functionality. And so a lot of these input products, a lot of management practices, anything that you're going to do that's going to change the soil biology is going to change the functionality. And the cool thing about the Biome Makers BeCrop technology is that we can really kind of point that out fairly specifically. And I've been working with Biome Makers for about 8 months now as an agronomist.
Great. Yeah, so I am Devin Clarke, and I am the permanent crop manager with Yara North America. And so my role is kind of threefold in many cases, really looking at supporting the commercial team with market development related activities, being kind of the day-to-day contact to be able to troubleshoot some technical aspects of nutrition management and just overall permanent crop management, as I did spend about about 5.5 years as an advisor in the field previously, and providing that support again to the commercial team here. And then I also oversee a lot of our permanent crop related R&D efforts locally within the state of California. Definitely have an affinity towards tree nuts, and had a lot of experience in tree nuts prior to coming on board with Yara, and so that's a big strong point in my background. So, the reason that Biome Makers has included me on the panel today is that we conducted a trial with them last year at our incubator farm in Modesto, California. So, to provide a little bit of historical context here and background, the incubator farm is something that we launched. We're on our third full growing season now, but there is an agreement that was signed in 2018 to lease a commercial production orchard, and this is in the Modesto area again. And when we took over that lease, we also redesigned the irrigation infrastructure so that we could run 4 separate nutrition management strategies simultaneously through the growing season on 27 acres of this 40-acre block, and do that in a replicated and randomized fashion so that we have statistical power in any of the results that we were able to generate from that. We decided early on to take more of a crop solution approach as opposed to a product specific approach, and look at what the conditions of that site were, what the challenges were, and figure out ways to more accurately address some of those challenges with these varying levels of crop nutrition solutions. So, with that being said, some of the key challenges that we faced at this particular block would start with the water itself. I think was the biggest challenge, and so our water source was a well because of challenges with district water infrastructure. There is access there, but not for low volume applications, only for flood applications. So that well water, we had about 1,100 pounds of bicarbonates per acre foot of water coming in from that water source, which created a significant challenge for us in managing overall EC, which was about 0.98, and being able to maintain a healthy soil environment for our orchard to thrive. So that was kind of the main challenge when we took over this block. And with these crop solutions, we're really looking at ways to provide a greater breadth of information to growers and to advisors as to how these different nutrition solutions react or perform in a real world setting, right? Where you're dealing with Mother Nature, where you're not controlling for every single variable as you would in some academic related trials. And so there was this interest in looking at as many different metrics as possible along the way that are not only fundamental for growers, advisors, and processors in what they're interested in and what they get paid on, but also in kind of pushing the envelope for almond production and understanding how different management changes can impact physiological aspects, stress reaction aspects of these trees, yield quality, ROI, and then also looking at the unknown, which leads us kind of to the trials that we were doing with Biome Makers, and that being the soil interactions. I think there was a lot of, myths that I was interested in displacing when we had connected with Biome Makers and learned about the technology that they provide as a solution to the industry. And some of those, as my days as an advisor in the past, that you came across were that mineral fertilizers are a problem, right? That is a problem for food production, for sustainability, for longevity of our soil health in the agricultural industry. There was one. One that I also heard frequently was that nitrates, specifically, nitrates make soil biology lazy. That's one myth that was commonly thrown around, because the fact that there is no conversion needed to get nitrates into the plant as there would be for, say, urea or to some degree ammonia, depending on the plant species, right? And that, to me, it was really trying to display some of these myths and better understand what interactions were actually taking place below the surface where we can't measure these physical, tissue sample levels or visual observations or anything of that nature to understand how these different programs, these different inputs, and these different management strategies are impacting that soil biology. Because that soil biology really is critical as a building block for all of the downstream processes that ultimately relate to yield and productivity, right? So with that being said, there was what we did at the trial last year was we were comparing we had 4 different treatments, as I said. They are kind of stair-step in their attention to the site-specific conditions. And so what we wanted to do is, just as a way of dipping our toe in the water, was to look at a comparison between the My Almonds crop solution, which was kind of our ultimate, throwing everything at the kitchen sink at it and using all the tools and resources available to us in the industry to make better decisions, and compare that against the baseline program, which is characteristic of a lot of growers still today in the industry, where you're kind of doing this 4-slug mentality, calendar type of application, about 50 units of N via UAN from March to June. And so the difference between that baseline program and this My Almond solution is that we adapted our input selection relative to the site-specific needs. We had more calcium applied, we had more potassium, or not potassium, we had more phosphorus applied. We had primarily nitrate-based applications with some calcium ammonium nitrates early in the season and periodically throughout the growing season for various reasons. But the biggest change is that we adopted this high frequency, low volume strategy, where we were applying, and it's not as frequent as some, but we were applying on a once a week basis from essentially leaf out until actually late July, if not early part of August. And that is very different from what the industry norm is. The industry norm would be more of a June 1 cutoff, and again, in this kind of calendar-based 50-slug application methodology. The reason why we adopted that strategy is because it more closely aligns with the most recent documented crop demand and uptake curves produced through UC Davis. And what we're trying to do is produce this kind of on-demand type of feeding management strategy, so that we're only giving those plants what they need and no more, and being able to optimize our input use efficiency in that way. So with that being said, beyond we're looking at, like I said, a lot of different metrics. We're looking at elements like nitrogen use efficiency, water productivity, yields, ROI, carbon footprint as another one, nut set analysis, and things of that nature. And some of those, I think that some of the results that we were able to achieve through the BeCrop analyses are reflective of the mechanisms and the changes that we recognize in the soil through the analyses. So the other part that I want to mention too is that we did these BeCrop analyses multiple times through the growing season. We started with a pre-treatment measurement, and then we did 3 additional measurements throughout the course of the growing season. And we wanted to be able to use that structure so that we were able to identify what were maybe some transient types of impacts of either abiotic conditions or the applications of fertilizer itself, and then also be able to identify what are more trends. And that's something that, you know, we were able to clearly identify through the BeCrop analyses with these multiple timestamps, that there were some elements that were, you know, occurred at specific periods in time, and there were some elements that continued to show improvements over time throughout the course of the growing season. So with that being said, you know, some of the key takeaways from the reporting that we were able to get back through the BeCrop analysis during our cropping season last year was there were some surprising results, right? So I'd say first and foremost, especially with concern to the kind of economic and market conditions of this year, we saw increased solubilization of phosphorus and potassium in those soils. That has, you know, potentially significant implications for growers, specifically in a year like this where inputs are through the roof and crop prices are low. But, you know, that increased phosphorus can improve the overall metabolism of these plants and allow them the currency they need to be able to move carbohydrates and everything through the plant, and be able to fill the nuts, be able to manage some of the, you know, photosynthesis and transpirational activities. The potassium itself definitely supports water balance, and it likely has some implications for stress mitigation in these plants. And, you know, when you think about from an economic standpoint too, if you are in a K fixing type of environment, right, where we have some kaolinite type clays or something of that nature, this ability for the management of the nutrients, right? A lot of this just comes back to the management, the ability of that management strategy that can be adopted by really any grower that has the ability to inject fertilizer at all. The ability to move that to a high frequency, low volume strategy is also there. But if you can increase the solubilization of the potassium that may be locked up in some of those clay platelets, then you are able to more effectively take advantage of every dollar of inputs that you have applied to that field, and be able to meet that demand of that crop without sacrificing any potential yield loss or adverse reactions from, again, a stress management standpoint. The other category, the second category that we recognized a significant improvement on was the hormonal production. And so there were a few key hormones or pathways to these hormone productions that were measured through the BeCrop analysis. And that was, if I recall off the top of my head, abscisic acid, gibberellic acid, and auxin production. And so when we think about those elements, you know, I think about those particularly earlier in the season when we have a lot of activity taking place within that plant, within the developing flowers, within the actual, you know, cell division and elongation components of early nut set, right? And, you know, I don't think that it's unreasonable to think that this management strategy that we have identified that had specific and notable impacts on those three areas potentially is a good reason for the 11% improved nut set that we have seen on that My Almonds program compared to that baseline on average for the last 2 years. And so it's these, you know, there's just these mechanisms that we haven't had, you know, any kind of line of sight on it as to why this is happening, just being able to note that it is happening. And so we continue to uncover additional ways of detecting the mechanisms behind this. And so that's where the BeCrop analysis has been really helpful.
Hey, Devin, let me ask a question real quick. Did you find that overall we're not really considering the fact that, because we've never had a tool that could ever really solidly look at those indexes that you just mentioned?
Sure.
That the effect of fertilizers isn't what we're you know, the negative effect that we would say of how they affect the soil biology. We're not here to condone the fact that you shouldn't use fertilizers. We're saying, you know, using fertilizers in a way that's going to best be utilized by the plant is what we really want to focus on in this trial. So,
yeah,
you know, we do see some great positive effects when we looked at the rates and the application method and the way they're applied.
Yeah.
Did you see that as well? in the overall view?
Yeah, absolutely. I mean, this is it's a consideration that I've been working through for quite some time around kind of market sentiment and mineral fertilizers being, again, a problem, right? So a lot of the sentiment comes from the fact that mineral fertilizers are all salts to one degree or another, and salts are considered to be historically considered to be detrimental to soil health and soil biology and soil structure and everything that props up our plants to be able to be productive, right? At the end of the day, though, it's important to recognize that salts are electroconductors, right? And that's so when I mentioned the EC of our water, we had a 0.98 EC, and electroconductivity is necessary from a physics standpoint for transfer of energy, right? And so if we think about water conditions typically, or water sources, and we think about the fact that just broadly speaking, clean water tends to take things with it, and dirty water tends to leave things behind. It's always been kind of a discussion point that we've had with a lot of growers. And so you do need a nominal level of EC to have healthy, thriving in soil environments and to be able to support plant health. And so we just need to be cognizant of how these fertilizers, these rates, and this utility of the fertilizers that we are applying to these systems impact that greater soil environment, and make sure that what we are applying is done in a way that really, at a basic level, complies with kind of the practical implementation of the 4 R's of nutrient stewardship.
Right.
And if we do that, we can, and we have demonstrated here that in a lot of ways we can actually improve the overall health of the system, and we can actually improve the use efficiency of everything that's being applied. And there are actually studies also that have shown that a proper and intensive management of mineral fertilizers actually produce a more negative carbon footprint than organic production in a similar system.
That was what I was going to reiterate, was that we found in the study that the actual use of light rates of fertilizers can actually improve the soil biology. And so, Jacob, would this be a good time to probably look at the findings we found? Devin, would that be okay to incorporate that now in the presentation?
If I could, just real quick, Aldo, I should clarify. The reduction in carbon footprint I referenced was actually relative to conventional fertilizer management strategies, kind of a 4-slug calendar approach, not organic. My apologies there.
Got it.
Yeah, let me bring up we kind of did a summary of the report that we sent to Devin, and I'll kind of maybe walk through and show actually some of the detailed views that we had on this. Let me see if I can just share my screen here really quick. Let's see. All right, are you seeing my screen here?
Yes, we can see it.
Okay, so this is looking overall at this this program that Devin was talking about. So really what we did is, so here we've got kind of the experimental setup. Don't really worry about these numbers. These are just internal numbers to help us know which samples belong to which. But really what I want to show here is that we've got there are several different time points, and then we're having a non-treated versus this intensive program. So this, if I understand correctly, is kind of that baseline that Devin was talking about.
That is correct.
We're really looking at that baseline versus kind of that spoon feeding type of effect that Devin was talking about. And so we've got one that now usually when we do this, and Devin, you'll have to correct me if this is incorrect, but usually our time 0 is before you've done any of those kind of treatments. I don't know if that's the case here.
In this case, that was accurate, correct.
Okay, so really these ones are both nothing has been done to all of these samples here at time 0. Where we'll start to see, potentially differences are where we get time 1, time 2, and time 3, where we'll actually, really start to see any differences that the practice itself would have. These two are just showing us the difference between locations. And so I'm going to kind of move forward a little bit and show some of these. So this is kind of the way that we've displayed them in this case. So we break down nutrient pathways, all of these pathways into very specific groups. And then what we do is we can kind of plot it out where, so the line down the middle here is basically looking at what, in this case, the baseline, and then the blue line on top of it is going to be however that kind of the spoon feeding approach does to it. And so if it's a straight line like this, basically we're seeing that it has no effect. So there's no difference in any of the carbon pathways based on kind of that intensive that spoon feeding that we had. We can start to see there's a little bit that we see a little bit of a difference here, but it's really not significant, anything that we would really worry about. So this is looking at carbon, and again, we're only going to be looking at times 1, times 2, and times 3 across this, because time 0 was just looking at spatial differences, and we're able to capture that within here. So again, nitrogen, we started to see a little bit here at the end, and I want to point out something important here, and that's that we're not measuring nitrogen itself. So we're actually measuring the pathways that are responsible for moving nitrogen around. And so we're looking at, inorganic nitrogen release, the pathways that are responsible, the microbial pathways that are driving the cycling of nitrogen in these different areas. So, for example, when we get down to phosphorus and potassium, like Devin said, we start to see something pretty interesting here. And that's that over time, so remember we've got time 1, time 2, and time 3. Over time, in both the organic or the inorganic phosphorus solubilization as well as potassium solubilization, we start to have a greater and greater increase in those pathways. Again, this isn't looking at the soluble phosphorus that's in the soil itself. It's looking at those pathways that are driving this. So this is the biology that is saying that there is more that's going on. There is, we've got a, 63% increase in organisms that are capable of solubilizing phosphorus and potassium in the system compared to that baseline treated sample that we had.
And Jacob, I would add too, just for context also, that from a nitrogen management standpoint, the units of nitrogen applied to these treatments were essentially the same. It was just the way in which they were managed, right? And then also with the phosphorus, any application of phosphorus, which was about 10 units in total, P2O5, was applied early prior to T1. In the sample. So the effects, as you mentioned, these are really looking at the changes in biology in response to their environment, and not looking at the actual P that's really available in the system per se, from like a soil sample standpoint.
Right. And although I would hazard to guess that here you're going to have a much healthier system where you are actually able to get the microbes to basically solubilize a lot of the phosphorus that is bound.
Correct.
And
so an expectation that what was applied, prior to that T1 point in time, that you have a greater ability to access that phosphorus, which phosphorus is historically probably one of the most challenging nutrients to get actually into the plant, and so this is a big improvement for us.
Right. Yeah. Thank you. And so here's one kind of key takeaway that we were able to show. This is just kind of looking at kind of what those effects look like between that T3 of kind of the baseline versus kind of what we've got here with this more intensive treatment, and we're able to see really that we are starting to push this quite a bit. And I mean, in this case, you know, if you're just looking, you know, numerically, you know, you're getting quite a big difference in what you've got here. Some of the other things that we're able to measure, a lot of the minor nutrients. Now, remember, these aren't measuring the nutrients themselves, but they're measuring the pathways that are driving these nutrients. We didn't see anything here. I don't know that we would necessarily expect to see a whole lot there. And this one was interesting because we actually did see quite a bit of kind of these biocontrol pathways. Again, we're not measuring like active biocontrol products that are placed on the soil. We're measuring the biology. So we're measuring the natural system's ability to produce, you know, fungicides and bactericides, and
I was just going to mention some implications for that within an almond cropping system, specifically elements like leaf scorch and canker diseases that from a bactericide standpoint, if we could get some suppression there from those microorganisms, that's really valuable because there's not a lot of options for control in those areas. And then similarly, with invertebrates like Oriental fruit moth or peach twig borer, or even mites that may overwinter in the leaf litter, I would expect that there could be some implications for management of that with the increase in biological activity around these insecticide agents.
Right.
Yeah, exactly. And just to kind of show kind of an example of that, this is Bacillus thuringiensis. We're actually able to measure kind of the changes in that, and this dotted line here is basically saying this is what that baseline treatment looks like. And so the blue bars are basically representing the difference between that baseline and that intensive treated. And so we're seeing a fairly strong signal of Bacillus thuringiensis. Now this isn't like BT as in the you know transgene. This is the actual microorganism that comes from. And so this is the way of nature kind of taking care of it. So this is one strong indication that you are having a healthier soil that is better able to combat some of these.
And in this case, with the spoon feeding, this intensive program, we're able to actually grow beneficial organisms with that program.
Yeah, So I mean, also implications for NOW with mummy shaking, right? So if you're not able to get to the shredding or the mowing of those mummies at an appropriate time, and they are, you know, all over the ground where we have been able to build up these populations of natural BT. BT can wipe out NOW pretty damn well, and so the control that can be achieved with that, it's not to say that you want to rely on that, but it's nice to understand that there is improvement there, and that may provide some economic value to you.
Yeah.
Right. So I'm gonna move on to kind of the phyto hormones that you had mentioned a little bit. Now again, you know, my understanding is that you're not adding any kind of product that stimulates the phyto hormones themselves.
No, no.
So this is just showing how kind of this practice is influencing kind of the phyto hormones. And again, this is the phyto hormone pathways that are in the microbes. We're not measuring phytohormones inside the plants themselves. This is the pathways that are producing the phytohormones that are used to basically, you know, the microorganisms used to stimulate kind of that plant, that communication across the plant and the microbe.
Well, there's a lot of production of these phytohormones in the root systems as well, which is coincidental, considering that that's where these roots are in the soil and interacting with these bacteria and these organisms that help stimulate this production.
Yeah. Would you guys say Jacob, would you say the direct result of why we saw these high increases in the T1 and T2 had to do with the time of year? Devin, would you feel that as well? Do you think there's more production of those hormones in the spring?
Yeah, I mean, there's an element that is not to be lost here. That there's a lot of learning left to be done, right? And
All right.
understanding how we begin to change our management practices relative to the information we've been able to gain through this work thus far. But I do believe that there's far more activity that would be taking place during that point in time relative to auxin and cytokinin production with the bloom time period, with the actual fertilization of the embryo and the flower, all that cell division and elongation that's taking place during that time, as well as leaf out. So there's a lot of activity that does take place through that time period, and I would expect that has some temporal component to it.
Yeah, and I'm just going to kind of go back up here because I think there's a good point up here at the beginning that I neglected to show, and that's really kind of here where we're looking at so we're this was started in February. It looks like we're taking in March we're collecting time 1,
Yep.
May, beginning of May we're time 2, and towards the end of May we're at time 3. So this is, I mean, we're looking at kind of a good beginning of this season here. It's not like we're looking at the whole entire season.
Yeah.
So we are seeing quite a bit of improvement just in that short, relatively short amount of time. But it is interesting to see how these are kind of fluctuating, and it would be interesting to see how it fluctuates with the natural abundance of some of these phytohormones and when they're naturally high in the plant, and if this is actually somehow then kind of cycling back and contributing back to improved plant health. So basically by improving the soil health, we might be seeing a direct benefit of improving the plant health kind of at the time.
Right. Yep. Very good.
And then I think the last one that I wanted to show is just another where we've got a lot of the nutrient pathways that we're looking at. We're looking at phytohormone pathways, but we're also able to look at some of these stress adaptation pathways. So in this case, we've got a lot of these stress adaptation pathways. So to kind of clarify a little bit, some of these pathways have to do with biotic and some of them have to do with abiotic stresses. So some that we're able to measure, like salicylic acid, abscisic acid, those are fairly strong for kind of boosting the plant immunity. EPS as well as abscisic acid are also important for drought tolerance. And so, by measuring these, we're able to look at how well the microbial community is going to be able to help the plant, I guess, adapt or really kind of buffer the plant from a lot of stresses that might come through growth. And so in this case, we're seeing that there's a pretty good increase in abscisic acid early on at time 2. We've got, looks like, organisms that are going to help benefit the plant as far as salt tolerance. And like you said, a lot of these are provided as salts. So the fertilizers. So maybe kind of this spoon feeding is actually kind of priming the community to respond to kind of that salt a little bit better.
And I would add too, like in the case of the EPS up top there, and some of the salt tolerance aspects, because EPS is kind of some of the glues that help bind soil particles together to create appropriate structure. And the salt tolerance aspect, we did mention that there is a considerable amount of salt that is coming in from the water source, and due to, I mean, for me, this is a very strong and positive result, because due to that salt load that's coming into this system, and the fact that this intensive or My Almonds program has out yielded the baseline program and everything else for the last 2 years, the actual transpirational demands and water demand for that treatment has been higher, although the irrigation is something that the entire block is treated uniformly. So essentially every time, through every irrigation cycle, that intensive program is actually depleting more water from that root zone. And so we're actually, over time, because of the way that we are uniformly treating that soil from an irrigation standpoint, we're actually creating a bit of a detriment to that intensively managed program, because we're not able to adapt that irrigation to what the actual demands are, right? So, so something that we have documented in a tissue sample, in our tissue samples and our soil samples over the last 2 years, is that because of that inability to manage appropriately, we have seen a increase in the tissue level of sodium to about 2 times what the baseline program is. And we have seen an increase in the base saturation of sodium within the soil to about 2 times what that baseline program is as well, although everything started equal. But the interesting thing is that with the management strategy, with the improvements that we're seeing in the soil health that are documented here, we're actually at toxic levels of sodium, but we're still outproducing everything else in the trials.
Wow.
So I think a lot of that comes back to the adaptations that are being able to be made in the soil relative to improving that overall health.
Very good.
Yeah,
Jacob, was that it? Did you want to show anything else? Because we have a number of questions we need to talk about.
Yeah, I think that's pretty much the highlights of what we wanted to show with this trial. But it kind of shows that we're not just looking at what the microbiome looks like. We're looking at the microbiome in the context of how it's interacting with the plant, how it's cycling nutrients, how it's producing kind of these cross communication phyto hormones, and how it's interacting with the plant as far as stress adaptation. So it's really in the context of kind of that the plant itself, and how, you know, in this case, you know, a practice, but it could also be, you know, if you're looking at different inputs of saying, well, you know, I've got a different biological input or a biostimulant that I think is going to work great. We would be able to go through, and using these same kind of parameters, we're able to look and identify how does that biostimulant or that product or the practice, whatever it is, how does that impact the microbiome that then cycles back and basically benefits growth of the plant.
Very good. So Devin, I've got a few questions here that have come in, and I want to kind of go through these, and some of them, Jacob, I'll relay to you as well. First question is, did spoon feeding past June 1st into July, August have an effect on whole rot? And I don't know, we only went through May. I don't know if you went ahead and went past that in your trial.
Yeah, so we did, again, we matched up our applications with the crop demand curves that have been established through UC Davis. And this is part of what the Biome solution is helping people to recognize the resources that exist to make better decisions, right? So we are continuing those applications all the way out essentially until harvest, but we are still tapering. So we're starting out with larger applications at the early part of the season in congruence with that curve, and then tapering off through that later part of the season. So we may be doing weekly applications of up to around 20 units or so in March and April. By the time July rolls around, I think we only put on 20 units through the entire month of July, right? Okay. So with that being said, the whole rot situation with that spoon feeding strategy and carrying it out that long is actually one of the most consistent and surprising improvements. And so we have actually reduced the whole rot by 33% on average over the last 2 years. And that's not a wide variance. It's essentially 32% 1 year and 34% the other year. And so that reduction, I believe, comes back to the hypothesis from some of our academic colleagues that has yet to be validated, that when you are oversupplying nitrogen, we understand that nitrogen and irrigation water are typically tied to whole rot infection. But when you are oversupplying nitrogen, particularly in the early stages of development, as those cell walls are the cells are dividing and growing in those hulls, if you have created an imbalance at that point in time, it's not something that you're able to go back and retroactively correct, right? So you've created these weak cells with an improper balance of nutrition elements early in that growth stage of those hulls, and then once those hulls open up, they are already predisposed to infection. And so by maintaining a healthy level of calcium and appropriately balanced nutrition, especially through those early parts of the season, then I believe that that's been one of the main contributing factors to the success of the spoon-fed strategy, and not slugging on, 3 times what the plant needs at one point in time.
Got it. Very good. Another one, Devin, is you mentioned when we talked earlier about the water source, the 1,100 pounds of bicarbonates. Mm-hmm. Were all the blocks of the randomized trial using the same water and the same water quality across the board?
Yes. So to provide a little bit of context here, this is basically a 40-acre production commercial orchard, right? And we took 2/3 of that orchard because the back third was highly variable and not appropriate for a long-term trial. And so we took 2/3 of the front of this orchard, and then we just underlaid a lot of additional submains to be able to manage these different treatments. So really, what it looks like from an overhead standpoint is that every 2 rows is a different treatment, and that's being delivered through the irrigation structure itself.
Okay. Jacob, this one's for you. Can you really elaborate more on what you mean by a nutritional pathway? For instance, when we're talking about, in this case, you talked about the microorganisms and how we count those species to better show the functionality of it. Can you elaborate more on that?
So yeah, what we do is when we extract the DNA and we sequence the DNA, we're really figuring out who is there. So we're able to look at what organisms are there, and then that big database that we have that Al was talking about has a whole bunch of wholly sequenced genomes, and based on what organisms are there, we're able to say, okay, organism A that we found in our soil sample matches to organism A that we have fully sequenced in this big database that we've got, and it has genes for nitrogen fixation. And so we'll be able to say, okay, organism A went from, maybe there were 100 organisms to begin with, and then it goes up to 10,000. And so we see this big increase in organism A. That means there's this increase in the potential to fix nitrogen in that system. And so we basically then take all of those, there are a large number of genes that are responsible for each one of these different pathways. And so we kind of lump them together into kind of like the inorganic phosphorus solubilization pathway. And so that's several different, different pathways, a whole bunch of different organisms, but we kind of lump those together in our kind of proprietary database to say, if we see an increase in these organisms, then that means there's this increase in the potential pathways for the solubilization of phosphorus.
One more question. Yeah, that answers that for sure. One more question about the P and K pathways, which really were improved with this intensive program. Why are these similar? What is do the P and K solubilizers do the same functions both together?
Not always. So a good deal of the P and K solubilization is just due to localization of like changing the pH. So the bacteria or the fungi are able to secrete organic acids. And a lot of times in changing that pH, they're solubilizing, both potassium and phosphorus. And so in that case, it is overlapping, but there are some more specific cases where it's not just one or the other, or it is one or the other that they're targeting.
Okay. Devin, here's one. Did you use any other method to validate stress mitigation to ground truth of stress adaptation data besides the BeCrop test?
Yeah, so a lot. How should I put this? there's a lot of information that we are gathering through this process. We are keenly focused on the metrics that are going to have relevance for growers, processors, and handlers at this current time, but we're not losing sight of collecting additional metrics to glean information and look for additional exploratory areas, right? So with that being said, we have been doing aerial imagery, and we've been doing that and looking at the response using different indices, NDVI and MCARI and things of that nature, and looking at that from an individual tree standpoint, and then lumping those into treatments and being able to perform statistical analysis on those. And so we have yet to be able to really tie anything definitively back to specific management practices at this point in time, but it is something that we have collected data on and intend to do further analysis in the future.
Another one, Devin, because I don't know, did you do conventional soil samples on these trials? Because
we did.
A question here about base saturation increases with sodium versus the potassium and the other cations. Did you see any difference in that?
Yes. So so base saturation, we're seeing essentially reductions in potassium, increases in sodium, and a lot of that likely due to the again well, a lot of that likely due to the production on the potassium side that we're drawing from that soil from a removal standpoint, because we've averaged over I think over 2 years we're averaging 500 pounds or more per acre improved over the baseline treatment. And so we are extracting more potassium and exporting more potassium. And so we have seen that reduction. And then the sodium side of it, again, with a reduction in potassium, you're also opening up more exchange sites for sodium, and we have that additional water demand that's coming from that intensive program that we are not able to meet based on that need to manage the water uniformly across all 4 treatments. So in time, if we do have the capacity to install a variable rate pump, we would be able to then manipulate the amount of water being applied per treatment and be able to better address that. So it is a little bit of a challenge that we have to work around at this point in time.
There's another question about that soil mineral report. Is that you're familiar with the Albrecht method of cation balance?
Correct.
In higher frequency. So the question was, did you find any benefit of, because we're only looking at biology with our BeCrop testing, did you find any benefit of the soil mineral balancing with the reports that we offered? With
the reports that Biome Makers offered?
Yeah. Because I know we kind of went through that at the bottom. We didn't see much change in the biology associated with minor elements.
No, none of the minor. So the Albrecht, I mean, they're talking about the base saturation. So you're talking about your positively charged cations, your calciums, magnesiums, potassiums, sodiums, and hydrogens in most cases. And so really we've seen, I would say, aside from the sodium standpoint, which is a function of the water, I think that we've seen very minimal changes to the base saturation. We are applying significantly more calcium to the program, but we're also extracting more from a removal standpoint. But in all, I think the lack of changes that we have seen there is indicative of the value of this program to a large degree, because it's kind of reinforcing this theory of the fact that nitrates are not detrimental, and nitrates are not making soil biology lazy. We're seeing that there's actually with the input selection and tailoring that to the needs of that particular field, and the way that we're managing that, we're actually seeing these improvements overall in a lot of different categories in that soil health.
Very good.
Yeah.
Jacob, here's a question that when you were talking about the pathway results, when you're looking at these particular Phyto Hormones, the question is the transcripts of these enzymes in the soil, are we using a predictive model based on abundance of microbes? And I think that's the question here is that we're measuring, what are we actually measuring? The actual enzyme or the associated biology?
Yeah, so in this case, we're not measuring the transcripts, we're not measuring the genes themselves, we're measuring the organisms that contain those genes. So the idea is that, you know, if you have, so we're not looking at just the who is there, but we're also comparing what they're doing. So if you're just looking at who is there and you see an increase in, say, your, you know, Burkhold area or something like that that goes up, you know, that could be due to any number of reasons. But if you see an increase kind of across the board of, you know, nitrogen, you know, solubilizing or phosphorus solubilizing organisms, then there's a pretty good chance that that niche is being exploited by those organisms. That there's that benefit that goes along with it. So we have done studies where we've actually compared the approach that we have, where we're just doing kind of this, you know, we're doing the 16S or the 18S and getting that and then inferring that function and doing those models versus sequencing everything and looking at what we see there. And I mean, we're looking at about a 90% correlation between sequencing everything, which is cost prohibitive, and doing this approach, which, you know, ends up something that you can actually do on a pretty good basis.
Very good. Devin, we're getting closer to the end, but there's another question that you were talking about 11% increase. What was the ROI change from the base treatment versus intensive
Good
practice?
question. So it's important to call out that the My Almonds program, the intensive management program, last year and this year will probably be even worse, but it ran about 2 times the cost of what the baseline program was. It does cost more in a lot of cases to manage more appropriately to the conditions. So it was about $740 an acre, I believe, last year for this nutrition program on the intensive versus about 370, I believe is what it was. So for starters, just to calibrate that. But the overall ROI has actually been a 13% improvement over the average of the last 2 years. And so each year, though the cost, the crop price differed, and the yields actually per acre differed to a degree, the net increase in profitability actually ended up almost even, which was really surprising. And it was right about $750 an acre better for the My Almonds intensive management program versus the baseline program. And one other thing that I was going to mention too, I mean, we looked at the carbon pathways and all that kind of stuff, and carbon in California is inherently difficult to store just because of the temperate climate that we're in. But it's not. I want to make sure that there's no misconceptions that the ability to improve our carbon footprint is actually significantly improved with that intensive management program, with the improvements in the soil biology that we were able to document here, coupled with the increase in productivity relative to every unit of nitrogen applied, relative to every unit of water applied. We've actually been able to reduce our carbon footprint there by 20% on average over the last 2 years also, just by changing the way the inputs and the management of those inputs.
Very good. Well, guys, there's more questions, and we'll answer all these. We'll send these out to everyone that attended here. Jacob and Devin will see these questions and answer the ones of prayer that we couldn't get to all of them. But I do want to thank everyone for attending. We will make this available. Will be sent to everyone and will be available on our webpage within the week. I'm sure by the end of the week it'll be available to watch again if you want to come back and see some of the more intensive slides that we couldn't that we kind of went through here, and kind of go through it and look at things as you do it on your laptop or on your computer at home. So thanks everyone for attending. Devin, thank you very much. We really appreciate your collaboration with us in this, and look forward to future ventures and see what what opportunities may lay ahead in the future.
Absolutely. Thank you guys. I really appreciate all your efforts.
And thanks, Jacob, for your input. That's been very helpful.
You bet. Thank
Thanks
you,
all.
everybody, for tuning in.
Appreciate your time.